introduction, Joe.
Short is good. The shorter, the better.
This will be the introduction, and it's a great pleasure for me to introduce, actually, Dr. Josef Coresh. As you see, he is the George Comstock Professor of Epidemiology, Medicine, and Biostatistics, one of our great, may I say, founding fathers in epidemiology, in many senses.
George, not me.
George, yeah, well, actually, you're in his big shoes, and you're doing very well, I think. So, Dr. Coresh is very much in cardiovascular epidemiology, kidney diseases, but also recently, as part of ERIC programs, the atherosclerosis risk in communities program at Hopkins, in neurocognitive studies, wonderful, very good news for neurocognitive studies, actually. And you will speak about vascular disease across the lifespan. Beware of normal aging. It's a great pleasure to have you.
Thank you. Thank you very much, Bert, and thank you for coming and listening to this. It's been a wonderful day. It's been nice to meet with various people and learn about the wonderful studies you do here at Harvard and hopefully advance the public's health. So I chose kind of a broad topic which intersects some of the areas that I've worked with for a while. And I'm going to start with some sort of, this is going to be my outline, some definitions, talk about some age-related conditions and focus on blood pressure a little bit, talk about important vascular beds and results in the kidney, the brain, and the heart, and then make some conclusions. And in case you can't stay, I think the bottom line for me will be strive for optimal rather than normal. In that I think a lot of people too often use the term normal a little too casually, and it can actually be a problem. So that will be one of the basic things here. So I thought I would start with definitions, right? So I thought we'd talk about normal, so I went to the Merriam-Webster Dictionary online, and they say normal is conforming to a type, standard, or regular pattern. Normal working hours, he had a normal childhood. The effect of normal aging. I'm not cheating, that's what's there. So I'm like, oh, that's appropriate for what I'm saying. So what is the effect of normal aging? Relating to, involving, or being a normal curve or normal distribution. So we all know the normal distribution. And the problem is, when this is the normal distribution, right, the Gaussian distribution, Leon Gordis told me to use that term instead of just normal, and it turns out there's something in words. And the idea is that, yeah, things in the middle are more usual, maybe end up called normal casually, and then you might define things at the edges as abnormal. But then that assumes that everybody in the middle is okay. And we'll talk about whether that's a reasonable assumption or not. Then aging. The definition is to become old or older. He is aged. He grew more forgetful. So literally, that's from Merriam-Webster. You can tell how this is going to be very...
Epidemiology, that's built right into the definition.
Next, I'm getting all the way up to Wikipedia. Then we really get rigorous.
Oh, that's written.
So, then disease, a disorder of structure or function in a human, animal, or plant, especially one that produces specific signs or symptoms that affects a specific location. Right? So I think a lot of the talk will be, so is it normal? Is it normal aging, or is it disease? And it'll turn out that that terminology may end up dictating a huge amount of how society will treat something. Right? So I'm talking to Chiadi and Dumile, and he's talking about the fact that the AHA is trying to decide whether obesity is a behavior or a disease. Right? And that may relate to a lot of drivers of things. And certainly we know that being obese is not at the tail of distribution. The process of becoming older. So then I went to Wikipedia and I thought an entertainment slide would be, so what do they say about aging? Symptoms experienced by the majority of humans during their lifetime. Wrinkles. Okay. Fertility decline, menopause. Happens. Presbyopia, right? So farsightedness, fine. Gray hair, fine. Hair loss, fine. In some sense, a lot of these, happen. Then hearing loss. I actually added, that wasn't that, I can't remember, one of the two of the other ones I added that weren't in Wikipedia. Hearing loss, greater than 50% of people aged 75 plus years old. Actually, that was there. And so now that's the question is, well, that's interesting. Vision loss, cataract, right? Cognition, dementia. By the time you get to be age 85, the majority of the people have some form of dementia, right? Atherosclerosis, leading cause of death. Kidney disease, kidney function declines with age. This is a funny one. It has, after the age of 30, the mass of human body is decreased until age 70. Obviously, it was written at another time, right? So normal and expected and physiologic is context relevant, right? I added tooth loss, right? Because I think this is an interesting example. In some sense, I think the dentists are ahead of us because the teeth are so accessible, right? So I think the thing is, if you take what happens with aging to be normal, then it's normal to not be able to hear, not be able to see, not be able to think, have your heart not be able to function, your kidney not be able to function, and have no teeth. And in some sense, I worry that when people say that's normal aging, don't do anything about it, it's like, that's obviously a problem, and for me it's been a problem in some of our research. I think the tooth loss is interesting because, right, used to be you'd think of old people and the majority had dentures, right? And at least that's a fix for not having teeth. Then now we have technology fixes and implants. And then we really have prevention where now with fighting periodontal disease, you can actually get people to the age of 100 and still retain a lot of their teeth. And I think the question is, can we do this for all the other organs, right? So this is still in the entertainment portion of the program. So you know who this is, right? Jane Fonda. And how old is Jane Fonda in this picture? 81. Now, then someone said, maybe it's an example of plastic surgery. Maybe, but maybe we all need technology, you know? And AARP makes an amazing video about old age. I don't know how many people have seen this AARP video. Watch it. Look it up. It's wonderful. It's got millennials. What is it like to be old? And they describe it and imitate it. And then they bring in older people who can do yoga and dance and this and that. And the millennials are like, I've changed my definition of old. Of course, the teenagers, their definition of old is over 30, So the research stuff will come from the ARIC neurocognitive study for some of it, and these are pictures of real participants, sort of younger and older, and it's actually interesting to see the range of variation, and I'll show you some of the data. So then I thought I would start with blood pressure, right? So you know this, blood pressure increases with age. So this is an example from Ann Haynes, and you see systolic blood pressure increasing in age in whites and blacks. The definition of hypertension, it was 140, right? And then before that, people used to think, oh, if you were older, you needed more blood pressure to get up to your brain, and the definition was in the 160s, right? And so the question is, you can tell, if the mean is over 140, then the majority of the people are going to be in the abnormal category, in the hypertension category, right? So there's the question of sort of what does that mean? It's interesting. This is a paper by Vassan from the Framingham study. Cumulative lifetime incidence of hypertension is over 80% in the Framingham study for women over age 65, right? So hypertension becomes almost universal. Does that mean it's normal? And does that mean we should ignore it or not? And I think you know the answers for hypertension. This is another example of the slope in increasing of blood pressure is about 0.6 millimeters per year in most populations, right? So some people may end up calling it normal. Most places across the globe, that's what happens. Well, it turns out this is a slope of 0, and 4 populations in Intersalt had a slope near 0. So it may be that this is common, but other things can happen. So it's certainly not inevitable, and I think we should distinguish between sort of median and common versus inevitable, and maybe we shouldn't just give in to the common and seek some things that are unusual but may become more commonplace if we did the right things. And this relates to sort of Jeffrey Rose's classical paper. We make all the students read that for us. You may make all the students read that here. And it's interesting picking this quote. This is the blood pressure in London civil servants. This is in Kenyan nomads. And you can see that the 140 definition for Kenyan nomads was very high. But for, the London civil servants, it's almost half the population. So it really depends on where you are. And he says to confine attention to within population comparisons has caused much confusion in the definition of normality. Laboratory ranges of normal are based on what is common within a local population. What is common is all right, we presume. right? And then, that is the wrong presumption. And I'll show you various arguments in various organ beds as to whether I think obvious concepts, I think, have profound implications. This is a slide that Paul Welton has shown in our cardiovascular epidemiology class, and I've borrowed here, which is a known concept that if you look at the prevalence of blood pressure, the people with the highest blood pressure of 180 plus have the highest risk, but they are the least common, and therefore only 7% of the disease in the population is due to the highest risk. The greatest proportion of the population that's explaining disease is really in the middle of the distribution, blood pressures of 130 to 160, and that's the prevalence where if you go down to 130, now you've got, sort of half of the population. And in fact, it may not be a coincidence that somebody thinking like this, like Paul Welton, was co-chair of the guidelines, which then redefined hypertension to a lower cutoff. And now we have to think of how do we deal with this. And I think some people are very upset, and other people are saying, we are now telling society what you need to do at an early age to really get to optimal aging at older age, right? So that was sort of familiar stuff, and some of those slides were things I saw when I was a graduate student, which was a number of years ago. And then what happened myself as we sort of looked at kidney disease over time, and for me this journey is sort of from 2000 when we started preparing for the 2002 guidelines for the definition of staging of kidney disease, to then 2009 when we helped form a global consortium to study it, to the 2014 guidelines and subsequently. So when we entered this, some of the very fundamental questions about kidney disease weren't really answered. And I started my thesis with very nuanced genetic epidemiology questions, and then I found out that sort of mid-career, or whenever mid is, I was dealing with much simpler questions. And then I found out that the simpler questions and the simpler papers had much bigger impact, which was kind of interesting. And now I go between things that I think are really cool and things that I actually think are important. And every once in a while it's both, but actually, often some simple stuff is more important. So in some sense, I wrote down these three questions. How common is kidney disease? What happens if kidney disease isn't treated? And how do we improve treatments for patients with kidney disease? So if you want to know how common kidney disease is, you have to define kidney disease. And it may seem silly, but there were all these textbooks, and there were hundreds of different definitions, and there were hundreds of different things, and they just didn't align. There were sort of articles, chronic kidney confusion, which was some people called something mild, and other people called something severe, and it was the same thing. And sometimes the things that were called moderate were more severe than the things that were called severe, and vice versa. So how do you define kidney disease in a way that might go across all the different diagnoses and etiologies? So for that, Andrew Levy and others on the work group said, what do the kidneys do? The main thing they do is filter, and they actually get 20% of cardiac output, right? So, and they filter about 120 milliliters of blood each minute. So in many ways, when we talk about a vascular talk, this small organ is getting 1/5 of all cardiac output because it needs to excrete waste for the whole body, retain water and salt, get hormone balance, and regulate homeostasis, right? So the idea was that if one of the main functions is filtration, maybe we can anchor the definition of kidney disease around filtration. So then if we wanted to estimate filtration, we needed to do that, and I tried to get an estimate that would work, and Andy Levy had published the MDRD study equation. We applied it to N. Haynes, and the first time we applied it, the data from N. Haynes here line up relatively closely to the classic data from Inulin, from Davies and Schock in the 1950s. And it was interesting. There are not many studies of gold standard measures of kidney function in the population. So Davies and Schock actually in Baltimore did what would have been a meta-analysis, gathered all the data of people that they considered to be healthy, and these were the measurements. And you can see that young healthy people had a GFR of about 120, but then by the time you got to the 80s, their mean GFR actually was variable, right? Some people were really above 90 still. Some people were in the 30s, even though they were said to be healthy. And so the mean was around 60. So some people wrote for a long time that normal aging is that kidney function declines by about 1 ml per minute per year after the age of 50. Back again to like blood pressure. This is what happened. Well, what does normal aging mean, etc.? So we tried to get an estimate, and it turned out these are the estimates we got. And when we got here, I thought we were pretty close. Before that, it turns out serum creatinine, which was the anchor for estimating kidney function, was measured incredibly precisely. So the coefficient of variation was about 2 to 3%. But across different laboratories, the magnitude of the bias varied by as much as 30%. So it turns out some labs measured low, some labs measured high, and they didn't even tell you which was which. And it did changed over time, inpatient, outpatient, so nobody really cared. So it turns out if we were going to use this to anchor the definition of kidney disease, we needed to standardize creatinine, and we needed to get a good measure. So we got frozen samples, we shipped them to a traceable laboratory, we did all that stuff, and it seemed to align. And when it aligned, it sort of made sense that young, healthy people should have a GFR of about 120, and you see the 5th to the 95th percentile, sort of harking back to the normal distribution, was from 90 to about 120. So 2 standard deviations below normal was about 90. Right? So there was a temptation to define disease at 90. Then we said, well, what about more extreme? What if you defined it at 60? That's losing half of your normal kidney function. Maybe that would be reasonable. And then we said, well, really what we should do is look at abnormalities and see when abnormalities start to accumulate. So we accumulated some of the literature, looked at abnormalities, found that they accumulated below 60, and defined that as to be disease. Right? And so we did that for 2002. Fortunately for me, right, Andy Levy was the chief nephrologist on the work group. I was vice chair as the epidemiologist, and we decided, let's look at the numbers and see what they mean. Right? Because too often the physicians can do really clever physiologic things, but then if you apply them to a population sample like N. Haynes, they may not line up in the way that you think. Right? So I remember a Saturday morning phone call where they wanted to do 90, and I said, well, actually, do you realize how many people are below 90 given these estimates? And it was 60 million people. And they weren't quite ready for that. Right? So this is sort of the tip of the iceberg type of thing, which is our model for a lot of disease, right? And they started a lot of this with stage 5 treated ESRD. So when in the 90s I was sort of coming on faculty, Paul Walton told me there was an epidemic of kidney disease on dialysis. There were too few epidemiologists working on it, so I ended up switching from lipid to kidney disease. U.S. prevalence at that point, in 1972, Congress approved federal funding for dialysis, right? And I think there were sort of 2 tenets. One was that dialysis was relatively uncommon, which was true. This was a life-saving therapy, so having people who were wheeled up to Congress and said, they're going to die if they don't get dialysis, shouldn't you pay for dialysis? And they're like, okay, we need to save these people. And then I think the other part was, well, national health care is around the corner, so we might as well start here, right? So they started there, and then they didn't quite finish, you know, Congress. But it turns out the rare disease by 78 was 42,000 people, and now it's over 500,000 people that are on dialysis every year. So it's still rare for a population of about 300 million people, but these guys are expensive, so it turns out it's one of the leading costs to Medicare already, right? And then the idea was in the late 90s was, well, if this is dialysis, what is pre-dialysis, right? And I think in epidemiology, we should always be cautioned of thinking in time backwards, because time doesn't work backwards, right? It works forward. So pre-dialysis is a backwards definition. We needed to do a forward definition. So if kidney function, you know, starts at 120, and it turns out when you're below 15, we decided to call that kidney failure, and somewhere below 15 or below 10, you need dialysis or you're going to die, right? And so then we decided, let's define stage 4 as double that, 15 to 29. Figure out using N. Haynes and estimates of kidney function, which I talked to you, we spent a year calibrating about how many people had stage 4 kidney disease. And the answer was about 700,000 people. And I think that's kind of what they wanted. A little bit pre-dialysis, late-stage kidney disease, what happens to them? Since then, we've worked on risk equation. Turns out the majority of these people don't end up on dialysis. They die from something else. So even though they wanted pre-dialysis, they look right, they're the right number, but they're not all the people. There are only some of them that end up going on dialysis. So they're not all pre-dialysis. They may be pre-death, like the rest of us. But they are at very high risk for dialysis. So then the idea was, well, what about prevention? Maybe this is too late. You can get these guys prepared for dialysis. You can do care. You can do management. But to completely prevent, it's too late. So what if you went 2-fold higher to less than 60, where we showed there was a range of complications, et cetera? So we applied that to NHANES, and we got a number that they were a little shocked at, right? And the number was 15.5 million people, right? Or about an 8% prevalence of the whole U.S. population, right? And to Andy's credit, he said, maybe. I mean, if we showed that everything's consistent and the construct is related to risk and the construct is related to abnormalities, then we have to take it. But that really ended up being a transformative thing, because that is no longer the tail of the distribution. Now you're biting a much deeper bite. And then there was the idea of, well, if you were above that, we weren't going to declare that disease, but if you had albuminuria, which was a marker of kidney damage, then you had disease based on another marker, which was albuminuria, and that got you another 6 million and another 3.6 million. So all in all, by 2002, we had an estimated prevalence of greater than 10% and a prevalence of 26 million Americans. When the people who convened this, I think, were thinking, you're getting us pre-dialysis. There are about 600,000. We need more nephrologists. Let's get it standardized. Let's get a real definition. And let's get this, get more money, get more training, whatever. But what we came up with was 26 million, which is just totally different, right? So for us, it made sense. And a lot of people liked it for a while, and then there was some resistance that I'll tell you about. So to estimate the prevalence of kidney disease, the hurdles we had to overcome is we had to get a large nationally representative sample. And then it turned out NHANES did that. And they paid, $100 million to do it. But for us at that point, it was $20. And we had just gone from like Sudan to Stata. So it was really easy. And we could do things 10 times faster than NCHS. So it was really quite convenient with a master's level graduate student to do all this stuff. Then we had to convert serum creatinine to estimated GFR. We needed the calibration. That took me 2 years. We needed a good equation. Conveniently, Andy Levy had published the MDRD study equation in 1999, and we found a simplified form of that. Then people complained about that, and we spent a decade improving the equation. And then it turns out, I'll show you the improvements, but the people who thought that the first equation wasn't good enough, by the time we did it in 2009, said, not that big a change. We can use the old equation. And it was mostly right. Then we needed to use a program that made computation easy. And, I showed you the implications of really coming up with an estimate of prevalence of 20% is that CKD cannot be the focus of nephrologists alone. And depending on your perspective, if you're a broad collaborative person, you're like, this is wonderful. Now we need to talk to cardiologists. We need to talk to endocrinologists. We need to talk to family docs. We need to talk to geriatricians. We need to think about drug dosing. We can collaborate. Like, you took my disease away, and now you're like making it something big that anybody can deal with. What's going on? This is not kidney disease, right? And then we needed universal calibration of serum creatinine, because if we were going to use these equations and errors of 30% bias in calibration from lab to lab were acceptable, it wasn't going to work. To Tom Hastetter's credit, the National Kidney Disease Education Program took that on, met with the manufacturers, and it turns out the manufacturers said, you want it standardized? Just give me a guideline, tell me what you want it standardized to, and within a few years it was globally standardized, right? So it turns out you need to get consensus to make progress. And specialists sort of loved and hated the high prevalence of greater than 10%. And at this point, the global prevalence is estimated at 10%. By now, there's an app for doing this. But I actually think an app took like many years later. I think that apps in many ways are the past, not the future, right? The way we actually made progress is by getting clinical guidelines to say what we need and then getting automated reporting by the laboratories, right? Which in a way is trivial. On the other hand, I remember meeting with a leading laboratory expert, and the equation, the Cockcroft-Gall previous equation was really pretty, and you could memorize it, and it was 140 minus age over body weight. You could do it with a pencil, and everything was lovely. This has exponents and long numbers and logs. And I remember meeting with a lab person, and the lab person said, I don't know that our lab system can do exponents. And I gave him the same look. I'm like, what do you mean? It's 2001. Go back to your computer. They have to be able to do exponents. And he was serious because, the LMS has it's for certain things. But they got it. So we recommended they should all report it. By 2003, some labs did it. And by 2008, the vast majority of all labs in the US and now globally report this thing. And I think that's, for me, the future of a lot of the stuff we do. Right? The idea that you can have an app or a website to do calculations, I think is just silly. We've got computers everywhere, and they should just calculate all the things that are calculatable. And how we get there, and then physicians should intelligently use this stuff. But I think in some sense, at least, we were able to do this thing and get it to go across. Then when we did the better equation from MDRD to CKD epi, the overall prevalence difference became from like 13% to 11%, right? So some people could be disappointed and say, oh, you know, all this work, a decade, and you changed it by 2%, right? But for me, it's actually reassuring in that mostly it agreed in the disease range, and the problem was in the near upper levels of, see, I can't resist saying normal, upper level of the non-abnormal distribution, and so, yeah, I need a better term, huh? Closer to the mean. Or maybe we should be able to use the term normal and abnormal, but it's just that the cutoff of 60 is now based on an absolute risk-based physiologic cutoff rather than an age-specific percentile of declaring something normal because it happens with age, right? So I'm vindicated maybe. And then when you look at this by age, the younger age groups actually had a decreased prevalence. But the people who were complaining us at that point, which were really obsessed with the high prevalence at older age, it actually went up. And if anything, the prevalence was close to 50% of all stages of kidney disease at older age, right? So our concept, which was in the guidelines, was that really what you need is nephrologists taking care of severe kidney failure and severe kidney dysfunction, and then primary care physicians taking care of people in the earlier, and an interface in between. On the other hand, this was a 2008 editorial, and there was a series of editorial and some steam getting up on, people love debates, so they would invite us to do these debates. And we were like, oh my God, you know, just what we need, you know. Right? The question is, are you getting heat or light? So debate on the epidemic of chronic kidney disease, the challenge. An epidemic of chronic kidney disease, fact or fiction? Isn't that great? You work on something for like a decade, and somebody says, fact or fiction? Like, great. So they said here, a cutoff of GFR of less than 60 without any correction for age or gender, without any consideration of concomitant albuminuria, a substantial revision of the KDOQI and KDIGO guidelines for staging stage 1 to 4 CKD is overdue. Normal decline in eGFR with aging, right? So their contention was it's normal, you're inappropriately labeling people, and you don't yet have a cure for this, therefore it's a problem and you shouldn't do it, right? So what our response is, chronic kidney disease is common, what do we do next, right? And what we contended is that the full range of CKD has high numbers beyond the scope of nephrologists alone. So we contended, yes, if what you want is this narrow thing that only specialists take care of, this is not it, right? We need to refine risk estimates and distinguish between optimal and normal, right? So the idea is let's focus on risk. We need to target the most promising interventions for evaluation in clinical trials. There are way too few trials in nephrology, but the answer shouldn't be to give up. The answer should be, all right, let's do the trials and find out the right populations. We need to target treatments at the subgroup with the highest risk in which benefit is proven, and we thought it was premature to use the high prevalence or lack of proven treatments to define individuals with reduced GFR as healthy, right? So what happened? They wanted to have a KDIGO controversies conference in 2009 to sort of make progress. And to make progress, Andy Levy and I suggested that rather than having a fun meeting where everybody comes and talks like I am today and everybody comes out smart but nobody agrees on anything, we should have everybody come with data, right? And see what the data say. And then I realized that as we were on the planning committee, if you wanted to know what the data said, you needed to decide on an analysis plan. And if you really wanted to know how GFR related to risk in a nonlinear fashion, and you really wanted to know how albuminuria related to risk in nonlinear fashion, you needed to run some pretty sophisticated models. And I realized that I didn't quite know how to do that. But if I wasn't sure how to do it, if I asked everybody to do it in a non-standardized manner, it would never work. Now, conveniently, I had just been back into GWAS analyses. I think you may remember 2007, 2008, 2009. It was kind of the happy days of CHIPS and GWAS and meta-analysis and whatever. But then I sort of they got more to p-values and more technical, so for me it was interesting to move into another field. So I decided the methods of GWAS and meta-analysis were perfect for a non-genetic topic. And so we went back to the conceptual model which Andy Levy had developed. The idea is you go from normal to kidney failure to death, but it turns out, importantly, there are non-kidney complications. And we should acknowledge that a lot of people go through the complications to death, and the higher your kidney disease, in fact, you expected the risk of dialysis to go up, but also the risk of heart failure, cardiovascular disease, mortality to go up. So we can't just look at kidney failure. So we were going to look at the full range of things. So we decided the outcomes should be kidney disease-centric, kidney failure, decline in GFR, AKI, but also mortality and cardiovascular disease. And in fact, we defined a whole range of other outcomes we should look at. The risk factors should be eGFR and albuminuria, but in fact, you should consider all the covariates. And so what we did for the Controversies Conference is the goal was to characterize CKD prognosis through individual level meta-analysis using state-of-the-art methods. And so we invited cohorts that were general population, high risk, and CKD. And this was an interesting thing. In doing a meta-analysis, if you want to be a purist, then you get just the right kind of cohort, just the right same kind of methods to get the most uniform result possible. But here we had a situation where there was a controversy and people were saying our data disagree. So if you have a controversy, in some sense, you need to respect all the data and get everybody's data as much as possible. So we actually did the opposite and said, we'll take every kind of data possible and see how they line up. And in some sense, there was a risk that they wouldn't line up, and at the end of this, we'd find out that we were wrong and we should redefine kidney disease. On the other hand, it seemed like this was the only way to make progress, and it was going to be a lot more fun to look at the data than just stand around for another 20 years. And then we decided everybody had to have the key risk factors or else you couldn't run the key models, and the analysis was going to be a survival analysis, a meta-analysis, forest plots showing variation across studies. And it turns out we did all this, we did the analysis, and then they said, oh, you did a great job, now start a whole consortium and do it again. So we did it all again. And we ended up showing up to this with hardly any budget, within a year, with 2,000 slides and 300 slides of meta-analysis, right? Because we really needed to do this, and it was clear that you could only do it centrally. And then we published the first paper in Lancet and a series of papers afterwards. This is a slide acknowledging the cohorts that contribute, and really each of them has contributed. You know, you guys run cohorts for like a decade or more, and then meta-analysis people just kind of put it all through a machine and come out with some answers. This is Kuni Matsushita, who was a fellow at the time. And it was interesting that, right, when you're students, it's interesting to see what the opportunities were. He had just come from Japan, very talented, very good, and he applied for a small grant and didn't get it. So he didn't get the grant. I said, do you want to work with me on this consortium thing? We're supposed to do this. He said yes. He became co-director, and then he published the Lancet paper that then was cited over 1,000 times. So this is the main result. So this is the main result of the meta-analysis of general population cohorts. And what you can see here is that in people with low albuminuria in the blue, if you make the diamond the reference group at a GFR of 95, which is within the normal range, even though 120 is best for young, healthy people, and you did a nonlinear association with a spline, lo and behold, it bends at 75, it becomes barely but statistically significant at 60, and then the risk becomes higher for mortality. If you have microalbuminuria, or 30 to 300, or if you have an albuminuria greater than 300, you get this curve. And to me, sort of with a background in math, the fact that these things come out parallel on the log scale and actually kind of look this pretty was just like worth it. After a year of work and thousands of slides, it's like, this is great. You know? And then we did cardiovascular mortality, and by 60 you had elevated risk and a bigger relative risk. And then we did end-stage renal disease, and in fact, by 60 the relative risk is 4 if you have low albuminuria, 64 if you have high albuminuria. And this eGFR construct that a lot of people had criticized may be poor, but if it's 15, you have a relative risk of 8,000 compared to if it's 95 and no albuminuria. So even simple measures at the extremes are amazingly powerful, right? So we published that, and the idea was to sort of lead to things. If we wanted to look by age, less than 65, 65 plus, you can see that the relative risks of all-cause mortality were a little bit lower at age 65. So there's a hint of what was going on here. But the relative risk of cardiovascular mortality was almost similar, and I was a little surprised at that. And then we decided to dig in deeper. We looked at albuminuria. The relative risks by age were actually similar. And then we decided, well, that didn't fully answer it. It was pretty clear to me that we were okay by age, and we wrote the summary of the workshop, but we decided let's dig deeper, because there were people publishing that at older age there was no risk until very low GFR. And I was like, well, the data have to agree, so what is it that's going on? What's underneath that? So we worked on this paper with Stein-Hallen and published that one in JAMA. And if you stratify by age more finely and use as a reference group 80, and it'll matter where your reference group is, I'll show you in a minute. If you look at the reference age group 55 to 64, you get the lower the GFR, the higher the risk of mortality. By 60 you have risk, and by 45 you clearly have risk. If you look at the younger age group, 18 to 54, the relative hazards are actually higher, right? So you have multiplicative interaction with weaker relative hazards at older age. When you get to 65 to 70, by 45, the blue line is below the black line on the log scale of the adjusted hazard ratio. And these little points here show all the point-wise interactions, with 0 being lower than the black line and plus being significantly higher for the green than the black. When you get to age 75 plus, indeed the relative hazards are lower, right? So it's a little bit of a flatness, and then it rises. Yeah, maybe it's not at 60, maybe it's 58, but I think that wasn't what people were quibbling about. What happens is when you get to high estimated GFR, these are people with low creatinine. It turns out those people often have low muscle mass. So at that point, it's not just that they have these massive, great working kidneys, they also may have low muscle mass, which is a risk factor for other outcomes. So we should always be careful that the thing we measure isn't a perfect measure. And then it turns out if you change the reference point, for example, in the oldest age group, and used 100 as the reference group, then you could tell that the whole curve would shift down, and you wouldn't see any excess risk until a GFR of like 40 or 30. So some of the people who used over 60 as a categorical analysis were doing the same thing, and the reference group would shift up, and then the rest would get depressed down, and then the cutoff would look different. So this was where sometimes, if you have linear associations, it's simple. If you have nonlinear associations, actually modeling the spline, actually looking at the full distribution, actually thinking about putting your reference point somewhere in the flat part of the curve matters a lot. So I think there was agreement, but it was more nuanced. GFR less than 60 was associated with risk. There was a little bit of an interaction with albuminuria. And importantly, the question was, does this mean that then kidney disease is less important at older age, right? If you actually look at it on an absolute scale, the same model, the Cox model, can be applied to a single reference point at this age, right? And that's age 55 to 64. If instead you look at the age 75 plus, at a GFR of 80, the baseline risk of mortality is much higher. Instead of 10, it becomes 57, right? The relative risk went down, so it's less than 1.5. But the attributable risk is 27. Whereas in that youngest age group, where it was really dramatic hazard ratio from 4 to 13, so it's more than a threefold elevated risk, the excess risk is only 9. Which for me was, again, right, this is like epi, first year, attributable risk, relative risk, but I was like, 15 years later, I'm like, this is wonderful. We got all this data, we worked this hard, and it's actually working, right? So for me, it's like, yes, you have interaction on the multiplicative scale. Yes, it's less relative risk. No, it's not less important. Now the question is, can you fix it? Well, that's a different problem, right? But don't call it normal. It is not optimal. You would much rather get to GFR to age 75 plus with a GFR of 80 than a GFR of 45 or 30. So optimal is here at 80, staying near where you were before. And it could be that common things are not good, and we should figure out how to make them better, right? So that's sort of the kidney part of things. And the same thing happened for albuminuria with higher absolute attributable risks at higher levels. For ESRD, there actually wasn't that much of an interaction, so it wasn't as much of an issue. And then this became part of the guidelines, and the guidelines indeed stayed. And now 2 decades later, they're the same. The cutoff is still at 60, and it's still common. And the idea is that risk goes by GFR categories. Albuminuria categories now become a second axis for risk. And then both risk and frequency and intensity of encounters and visits are driven by this 2 axis of risk. Then the idea was that the kidney guidelines became clearer that the evolving importance of kidney disease, if it's that common, then it's not just a subspecialty. And strangely enough, the global burden of disease and other meta-analyses suggest that the prevalence globally is about 10%. And then we've got issues with poorer treatment and more accelerated issues in developing countries. We've got issues that now actually the burden of dialysis, there are more dialysis patients in Asia than in North America, right? And with economic growth alone of China and India, that number will double even if the disease does nothing, because just access will double, right? So there really is a recognition of what needs to be done. And then the consortium, you never get done. If you think you're going to put yourself out of business, never happens. So now we've got 80 cohorts, over 10 million individuals, and we're working on surrogate outcomes that we've had approved by the FDA to improve the conduct of clinical trials. So now a few slides on sort of other vascular beds. So this is the ERIC study, right? So we now have 10 million people in the consortium. ERIC, 15,000 people. We've been working on it for 30 or more years, and we're still working on it, right? So it's hard work, and it's good to respect that hard work. Aaron Folsom is the chair of the steering committee, and we're doing all sorts of neurological studies. So talking about dementia and the issue of what happens, this is midlife diabetes is a risk factor for cognitive decline, and you can see that if you have diabetes with poor control, you do much worse than diabetes with good control, with undiagnosed diabetes, with prediabetes, as a reference with no diabetes, right? And these data suggested that primary prevention of diabetes or glucose control in midlife may protect against later life cognitive decline. We showed that diabetes is a risk factor for dementia. Both prehypertension and hypertension are risk factors for dementia. Interestingly, if you image amyloid in the brain, very specific to Alzheimer's disease, vascular risk factors are strong risk factors for amyloid deposition. And if you look by ApoE4, people without ApoE4 had a relative risk, but actually people with ApoE4 had a relative risk. So this is, if you have the genetic risk for Alzheimer's, it's even more important that you prevent vascular disease because you have a higher baseline risk and even a higher relative risk, right? So this idea of we understand dementia, we really need to think fundamentally about what's going on, and we had more amyloid in African Americans, which is another interesting topic. This is another, again, the vascular health going across all beds. This is Life Simple 7, vascular optimal health, and the probability of good aging, short physical performance battery. So it turns out walking speed and other measures of physical function are also related to vascular health. So this idea that the vessels just take care of your heart, really it's everything and very broad as we think about it. Prevention recommendations, National Academy of Sciences talks about trials, but really talks about the idea that you may need observational epidemiology to make progress. We had the SPRINT MIND study, which is an amazing study, but showed no benefit of aggressive blood pressure control for dementia, but clear benefit for MCI and a composite outcome, right? So this JAMA paper is actually quite confusing in a way, but in a way there's a hint here, and the designers actually wanted to do better than they were able to do due to funding. The trial phase only lasted about 3 years, no difference on dementia. Then they extended a little bit to the trial and cohort phase, and they found a little more difference. And then you go further and you find a little more difference, but lots of uncertainty. And it may be that the observational data from ERIC NCS and other observational data suggests that you need to look at like 10 to 20 years, right? And it could be that it's not so easy to do trials. Never mind, we don't want to wait another 20 years after much of the epidemic and disease burden. So the ERIC neurocognitive study is still studying this. We're proposing an extension of new visits starting in 2020 annually to actually look at this in more detail and do more brain imaging with 1,000 MRI and PET studies. In 2013, I met Frank Lin, just a brilliant ENT surgeon, and he told me something that I didn't know, that hearing loss became common. I kind of knew, but this is like the hypertension thing, right? 80% of people over the age of 80 have significant hearing loss. And it's the kind of hearing loss where if you're a child and you have this, you need a hearing aid, of course. If you're older, I have a family member, you went to his physician, he said, you're just getting older. And the person's a leading professor. He said, just do this. Right? So I became convinced that Frank was smart and we should do something. So now we're about to finish recruitment for a randomized clinical trial of whether an intervention with hearing aids can remedy hearing loss and improve cognitive function and slow cognitive decline through decreasing cognitive load, changing brain structure, and reduced social engagement, right? And the neat thing is, it's an intervention, so there was the opportunity. And to me, it was like having done all this stuff with kidney, sure, I was declaring that it wasn't normal and it wasn't optimal, but like I didn't have a thing. Your ear, and maybe we should like pay for it and help people do it. Whereas right now, it's being treated a little more like a cosmetic and convenience device rather than a dementia prevention device. So that's it, sort of the last slide, and then a thank you. So I'd like to have shown you that vascular disease has important influences across all beds. The heart, MI and stroke, but also heart failure, the kidney with wide-ranging implication, the brain with an opportunity for preventable cognitive decline, and many studies by your chairman and others have shown a lot of this, and we're trying to contribute. Physical function, another opportunity. I think we should beware of defining what is common as normal, has been my theme for the day. And to a Department of Epidemiology, I believe epidemiology is powerful. I think it's perfectly suited for measuring risk. So this is where nobody can beat us. We can measure optimal versus high risk, right? Then we have to use other lines of reasoning to guide disease definition, staging, prevention, when combined with other lines of evidence. And in older adults with CKD and other conditions, I think shouldn't be ignored, and we need to rise up to the challenge of minimizing and reducing risk for patients of all ages. This is really team science. I work at the Bloomberg School of Public Health. We collaborate with medicine. We're in the Welch Center in between, cardiovascular epidemiology. This is the staff that collect the data at the Comstock Center. I work on the kidney disease studies with people at Tufts here in Boston, Andy Levy and Leslie Inker. And these are sort of the various consortia that we've worked with. And I like this definition of a consortium, a group formed to undertake an enterprise beyond the resources of any one member. So thank you very much for listening.
Thank you very much.
Questions?
Yes, please, questions. Yes, please.
Question. I mean, regarding the implications of this kind of thing to epidemiological research, just I'm curious, like, it sounds like we will end up kind of with some positivity issue somehow, or if we compare like young people and older people, somehow if they have kind of a similar level of any, for example, EGFR level or something, they're kind of like, it can be kind of extreme. Yeah, either older people or younger people can be from kind of the kind of extreme distribution, like the end. And somehow then what can we do with that?
Yeah, I think I get at some of what you're asking, which is if this sort of Gaussian distribution as you age gets lower function over time, if you get old enough, the tails of the young people and the tails of the old people may overlap less and less. And then it gets harder to say, is it possible for an 80-year-old to be like a 20-year-old, right? My argument is that actually too often we underestimate how wide that distribution is. We often look at confidence intervals rather than full distributions, right? So I had another slide that I dropped of 80-year-olds who've run marathons, right? And I can't run a marathon now, and I probably couldn't when I was 20. So there is an overlap, and we should use that overlap. We shouldn't overplay it, but I think we should not underestimate how dramatic some of the improvements are, and we shouldn't rule out what is reasonable for interventions, right? In that, I mean, through clean water, we've changed life expectancy, we've changed behaviors, right? I mean, car accident rates were so high, and through safety, seat belts, and various other rules, and now all the automated technologies, I think we're completely transforming that, right? What should we do for chronic disease? And when you look at blood pressure, can we figure out a low-salt diet that could dramatically change blood pressure in the entire population and then change the whole curve of aging, right? Or should we take on the polypill approach of saying, if we can't change diet, then maybe really everybody over the age of 50 really needs something that will counteract our sick population.
But you're hearing device, your hearing intervention is a different approach. It's not prevention, you're actually trying to treat a downstream effect of whatever causes hearing loss. Sometimes it's because you worked as a jackhammer expert when you were 20, but maybe it's hypertension, or maybe it's other things that are causing hearing loss. Is that an effective intervention to, I mean, how did you get that funded? Has there been proof that hearing loss leads to cognitive decline only because people are just socially detached, and that intervention works equally the same in everybody?
So the good thing is Franklin had been working on this for like 10 years,
Yeah.
right? And I just came in to help him with some of the
That's the
methods,
best time to
right? It's great, you know. And he was looking for the thing, and then it worked for him. So I got very lucky, and it was nice. They had shown in a number of studies that it's, first of all, very clear that if you have hearing loss at older age, you have accelerated cognitive decline and a markedly higher rate of dementia, right? They had shown that if you have hearing loss, you have changes in multiple brain structures, and they had hypotheses where because of the hearing loss and deciphering issues, the attention thing, like if you're on a noisy train or a noisy phone line, it's just horribly distracting, right? So there's this cognitive load, the idea that now you're out what people are saying. And then I actually think your other comment is relevant. Being in the school of public health, I used to think, well, it would be great if we just did everything by primary prevention. But I think now I'm like, all means possible, right? So I mean, right, we know the big problem is diet for heart disease and cholesterol, but it turns out statins are a good way to fight back. So it may be that maybe one of these days we'll figure out how to protect our cochlea, but until then, hearing aids are good.
Brings us back to Jeffrey, high risk and population. Joe, thank you very much.
Thank you.
See you here next week. Dr. Enrique Schisterman comes to talk to us then at the same time in the same place.